And we're back. So let's stay on this idea about these reactive oxygen species and apply it to another model. So Parkinson's disease, Huntington's disease seems to be affected by, the main symptoms seem to be produced by destruction of really neural terminals that contain dopamine. So what is it about dopamine that leads to somewhat of a neurotoxic output? So let's look at a nerve terminal now. We're going to be up in the cauda putamen, right? Part of the basal ganglia system. Hopefully you can read that. Okay, so typically dopamine, right, is produced in the nerve, synthesized in the nerve terminals. You have tyrosine hydroxylase coming down, and it gets packaged into a vesicle that contains dopamine. So you have these vesicular monoamine transporters. And you have, I'm just going to do this all in blue right now, you have the dopamine transporter. Right, so the typical process is the vesicle docks, dopamine is released, has its actions on the D2. I guess you can't really see that. These are the postsynaptic receptors. Right, so it has its actions there on the postsynaptic receptors, but that's not the main point here. The main point is now that dopamine gets taken back up through the dopamine transporter and it has to get repackaged. So it goes through those vesicular monoamine transporters and gets put back into the vesicles. And that is a very, very important step. Not only the removal through the dopamine transporter, but the repackaging. So that's what we're going to kind of look at in this respect, is that whole process here, that repackaging of that dopamine. Because dopamine, what's a good color for dopamine? So this is the problem when you have too many colors to choose from. So dopamine, that's green. Dopamine can be neurotoxic. So why is that? What does that mean when somebody says dopamine is neurotoxic? So what that means is, so let's look at a model system, and we're going to talk about methamphetamine as a model system when we think about vulnerability of neurons. So what methamphetamine does is let's make meth a different color. We have green, our dopamine. How does this one show up? Not very well. See, these are all live videos. You get to see all the fumbling around for different markers. Okay, so it's going to be purple, methamphetamine. So meth. Doesn't have to be all that bright. It would be nice if it was a little bit brighter. My children need new markers. Okay, so meth is purple. So meth blocks the dopamine transporter, right? It also impairs the ability of monoamine oxidase to have its action on dopamine. But the other step is that it induces the release of dopamine. So induces release. And this is a really important pathway in all of this. And it's not some pathway that's always that clearly laid out in terms of drugs and behavior or psychopharmacology. So what happens is methamphetamine floating out here in the extracellular space gets taken up by the dopamine transporter. So you've all heard that. So then where does it go? So in fact, what it does is, because meth mimics, so meth equals dopamine. Meth mimics dopamine. So it can get taken into that vesicular monoamine transporter. So now this is the pathway that oftentimes is skipped over, and we're going to go into this in a bit more detail, but I just want to lay this out now so you get some introduction to how these, the general common pathway in terms of neurodegeneration and these reactive oxygen species. So, and it doesn't only have to be produced by calcium. That's the main point, because the basal ganglia is a sensitive area of the brain in terms of neurodegeneration. So when you're looking at Parkinson's and Huntington's. So why is that? What about dopamine? Okay, so when methamphetamine gets into the vesicle, it competes with dopamine. And in fact, meth can reverse these pumps, this vesicular monoamine transporter, and start pushing dopamine out. And that is bad. So what does the cell do? First of all, the dopamine transporter starts working in reverse, which, right, you've all hopefully heard about. So now the dopamine transporter is working that way. So it's taking this free dopamine and shoving it outside into the cleft, okay? So that's what gives meth that extended high. I guess that dopamine should end. Oh yeah, that dopamine is green. Okay, so there's a lot of dopamine. But dopamine, right, let's go back to this. Dopamine is neurotoxic. It's neurotoxic when it's free floating. Notice it's not in the vesicle anymore. When it's in the vesicle, it's protected. When it's out here, it's not protected. So what you need to have happen when it's free floating is you need those enzymes. You need monoamine oxidase, you need catechol-methyltransferase. So you have those enzymes, right? So let's say this is inside. You have monoamine oxidase. So that's the problem with working with markers is you can't erase. And you have catechol-methyltransferase. So monoamine oxidase, catechol-methyltransferase working to break down that extravesicular dopamine. That's a mouthful. So outside, so let me just write it up here. Dopamine outside of the vesicles. That's when it becomes neurotoxic. So why is it neurotoxic? Because you're activating monoamine oxidase, catechol-methyltransferase. As those are working, lo and behold, what do they start spitting off? Reactive oxygen species. Let's change our color. I'm going to have to, before we do the next video, I'll have to make sure my colors are all proper. Oh, that's not bad. Reactive oxygen species. Reactive oxygen species. Reactive oxygen species start building up in that terminal. When they build up in that terminal, right, that can cause lysis. They're Navy. Lysis. So those terminals, those dopamine-containing terminals, will start dying off. And don't forget, there are mitochondria down here. So those reactive oxygen species can actually start affecting the mitochondria as well. They can start feeding in the mitochondria, right? Start working on ATP, and they can send signals like cytochrome C back to the cell body and can actually destroy the cell body. So just to make one thing clear is that this is a proposed mechanism by which dopamine containing terminals start to die off in Parkinson's and Huntington's. It's not absolutely clear, but that's why I present it in terms of these models, in terms of methamphetamine models, in terms of other drugs that seem to increase extravesicular dopamine within that terminal, leading to the activation of these enzymes within the terminal, monoamine oxidase, catechol-methyltransferase, leading to reactive oxygen species like hydrogen peroxide, so H2O2. That's a reactive oxygen species that can lead to, or these oxygen radicals, hydroxyl radicals, things like that. All of those working together, producing lysis of the cell terminal. And that can then destroy that cell or lead to destruction of that cell body and all sorts of other problems, like the generation of misfolded proteins, which is where we're going to go next. So I'll let you enjoy that picture for a moment, take it in. I'm going to pause and we'll come back with protein misfolding.